So I usually get invited to things in order to talk about this thing called substrate independent minds, which is what we call the objective of being mind uploaded, taking your mind and putting it into something else. But before I get into all that, I just want to say that I was fortunate enough to be at the first XFF, and being there and seeing what was going on there made me come back, and it even made us, a tiny minor sponsor of the event. The reason is because for me personally, this concept of combining art and science, that's really what it all goes back to. I mean, the reason I do any of what I do, the reason I became a neuroscientist is because of art. So when I was little, I loved to do a whole bunch of different things, and in fact, it was sort of music, writing, whatever, you name it. And it just seemed to me that there was so much, I could never get it all done, and there were a lot of things I couldn't even do. And just thinking about that, about my capabilities, about time, about stuff like that, that got me interested into the rest. That got me interested in science. So for me, that's really what it goes back to. It's all about being creative. So, with that said, I thought that instead of rushing straight to what everybody probably wants to know, which is, okay, so what is mind uploading? How can you do it? How far are we? Is it really possible? That sort of thing. Let's have a little look at, because we have this opportunity, let's look at apocalypse. The reason being, of course, that yesterday was supposed to be the end of the world, and this talk was supposed to be given yesterday, so actually I sort of had that. Oh, not this one. Where is it? Here we go. This was my slide originally, but since I got rescheduled, we're just going to have to hope for another end of the world today. See if that happens. Now, of course, that didn't pan out. And why? Well, I don't know. Something about the calendar didn't quite work out. But I thought I'd remind you all of the fact that, of course, there are things that actually can be dangerous. There are things that can be dangerous to the world and to humanity, to civilization, not just to lives as such, but the way that civilization is put together. And just to give you a little taste of that, I hope this works. Oh, this is a little video of what happens when actually there's sound that goes with this. Is there something we could plug in and just run this again? If there is, that'd be kind of cool.
It was. It's done. That looks right.
Yeah? Let's put it on this side. Here we go. I don't know if the sound is on, but maybe it is. Let's see.
A giant lump of rock heading perfectly through the solar system at 45,000 miles an hour. It melted surrounding rocks and sent molten bullets flying out at supersonic speeds. Superheated 2,000 mile an hour winds raced from the impact site. This was hell on Earth. First, a superheated fireball, then a mega tsunami. Nothing within the impact zone could have survived the onslaught. Just minutes after the impact, the Earth was ablaze.
So the point of this is that unlike the apocalypse of yesterday, this sort of thing actually happened, and it will happen again. It's just a matter of time. So there are real apocalypses, but those aren't the only ones, of course. We've got all this other stuff going on, like disease, violence, and then the ones that are a little bit bigger, the ones where we think maybe this is going to wipe out all of civilization. This is back from when people were worried about nanotechnology creating gray or black goo that would spread across the earth, basically turn everything into some substance. Then there's this old one that we hardly ever talk about anymore because, you know, the Cold War's over, but the stockpiles are still there somewhere, so basically it's still possible. This one's very popular right now. Biological warfare as a possible sneaky way of attacking an enemy and causing untold devastation. And then, among friends of mine, this is a very popular doomsday scenario. As we go along and we create our machines that become ever better and help us out, and ultimately, suddenly, it's like we're not the prime species anymore, and we're kind of at their mercy. That's a possibility. But really what it all comes down to is just that we're currently really engineered for a very specific niche. And I think that's what I wanted to start out with, because before getting into the technicalities of what mind uploading is about and how you do it, you need some motivation as to why would you even care about this topic? Why is that important? Why don't we just, you know, let's be happy with what we've got. Our bodies are wonderful. They're great. I love my body. And, you know, we could just extend our lives and be healthy. This isn't just about life extension. That's what I wanted to say. There's more going on here. What we have is we've got millions of years of evolution that have made us made us well enough adapted to survive on a planet that has certain criteria. Anytime something doesn't fit those criteria, we don't survive. And it's really not just about surviving in different environments. It's not like, well, what if it gets too warm, or what if there isn't any air? It's about other kinds of challenges as well. So when you think back to that image about the AI coming along, it's the notion that perhaps there are other kinds of challenges out there that we haven't faced millions of years ago, so evolution didn't make us well suited to survive that kind of challenge. Just because we're well suited for now doesn't mean we're well suited for everything else that may come at us. So that's one aspect, it's the survival aspect. There's more to it though. Well, let's get over this. There's more. Yeah. So there's a personal side to this as well. So this was about humanity before, it was about what if something could wipe us all out? How do we make sure we're not just stuck on one tiny blue ball that we can survive on? Where could we go? There are also personal issues. This is where, for instance, suffering comes in. The fact that we do all still die, that we slowly decay and become decrepit. Even if you think it's perfectly normal, if you think, well, this is just the way things always are, people have to make way for the next generation, it doesn't mean that there isn't suffering involved. And on a personal level, if you could help someone not go through a disease, you could help someone not suffer Alzheimer's, you have a moral obligation to do something about it. Now, this is where I would say that merely dealing with anti-aging falls short in a number of different respects. So one of them is that it doesn't help us deal with changes in the environment, changes in our challenges. It doesn't help us deal with the limitations that we currently have. And sometimes that's not just about surviving. Sometimes it's just about the things that we want to do. When I started this talk, I said for me it all began with creativity. So sometimes it's just because you want to be able to do more. Why should we be so limited? So if we create an artificial intelligence, for example, it can do things that we can't. Something that can remember everything perfectly, or something that can think faster than we can, or that can use, say, quantum computing to think in ways we couldn't even conceive of right now. Then the question really becomes, well, why do you just want to make that and be a bystander and see this go off into realms that we can't even imagine, developing wonderful new ways of thought and creativity? Why not participate with that? Okay, so let's get past all the traumatic things in here. Move on to the part that I think everyone's really more interested in. So, substrate independence. Why do I call it that? What is it really? Well, I think after what I just told you, it should be fairly clear that one of the problems is that we're stuck in a very specific type of body, of biology, and this is really what we have to deal with, and even with anti-aging, etc., that's the thing that you have to fix. But before we can talk about why would you then do something like mind uploading, let's think about who we really are. Let's find out what is it that we're talking about here. I'm going to skip over some more stuff that was talking about that whole selection thing I already told you about, evolution. I should mention that this used to be a 60-minute talk, but it's been crammed into 30 minutes now, so jumping over some stuff. So there's a concept here, system identification, which I'll explain in just a second, but it starts with thinking about who we really are. So, or what are we? We think that we're living in a world where we breathe air, we walk on the ground, we see things, we hear stuff. But what is really going on when you're doing that? When you think about it, if I'm sitting here and I'm breathing air, am I really breathing air? Do I notice that I'm breathing air? Or what's really happening here? Actually, there are air molecules that are going into my nose, and there are little hair follicles in there that sort of wiggle around, and then it causes an electrical signal to go up a nerve and into my brain. But I'm not even aware of that part. I only become aware of this once that signal is processed. So really, you're not aware of anything until something's being processed. You're not aware of any sensory information. You're not able to produce anything, to act. You're not able to speak, communicate with people. Basically, I'm not saying that the whole world around us doesn't really exist, but basically, you're only aware of it once things start getting processed inside your mind. So the brain, the stuff that's going on in here, that's you. That's who we are. Now, if you wanted to think about what that means in computational terms, well, it means that you've got a device that it's churning out results. There are signals coming in, and something is being done with that. It's churning out results, and you could say, well, how is this different from any other machine, any other device that is producing results like that? Well, it's not intrinsically different. It's just that it happens to be done with biological machinery. So how could we grasp that? If you wanted to, for example, carry out the same functions in something else, which you could. I mean, there are other types of substrates that do computations. What would you do? Well, right now, even if you think of this concept of going to a substrate-independent mind, and by substrate-independent, I mean something that can operate on a variety of different substrates, not, without a substrate, it's not a ghost. You could think about doing this in different ways, but the only way that we currently really can tackle, the only way that's feasible, is through an approach that we call whole brain emulation at the moment. And that's because it's a very conservative approach where we assume that you need to reconstruct the neurophysiology and the neuroanatomy to a great level of detail. And we have some understanding in neuroscience of how to deal with that, because for the last 100 years, mostly what we've done is we've been prodding around in pieces of brains and figuring out how do you measure something at the lowest level, at that detailed level where you look at the neurons and you try to get a signal, or we try to map out the anatomy. So those sorts of things we understand, even though we have no real idea of how the mind works top down. We have no idea what those strategies really are, of how, as we go about our lives, how do we carry out tasks, how do we make decisions, what is consciousness, what is perception, all this stuff we don't really understand, but we know somewhere this is happening down in that brain, and we understand something about dealing with those signals. So this notion we call whole brain emulation, bottom up. Oh, did I just go backwards? I guess so. Okay, now, sorry, I already told you this. Let's skip over that too. System identification. This is the notion that you have a black box, you don't really know what's going on in there, but you're trying to describe how it works. Now, we're initially saying that the black box is our entire brain. It's whatever's going on in there, but that's really complicated to figure out. In nature, we often have this, where there are lots of pieces of nature that, where there are different parts in there and they affect one another in some way, and we try to describe that by building representations and models. But if you try to describe it by looking at each piece in great detail, let's say, for example, here you've got a box with a whole bunch of molecules of gas in it, and you try to describe what's going on in that gas by looking at each one of those particles and how it moves, it can be very difficult. Whereas, of course, there are other ways of describing what's going on in a gas. So this is why the first thing you do is you try to make the problem as simple as possible. That's why physicists, as we see here with a big chicken, they love to talk about spherical chickens in a vacuum. That's kind of their approach to how to do science. And it makes sense, because you have to start somewhere, even when you make things more complicated as you go. In neuroscience, we like to look at effects that we call behaviors, and we're trying to describe what's going on in there. So the first thing when you're trying to do system identifications, you need to decide what parts of the problem are most interesting to you. So how are these pieces that have some effect that you're interested in, how are they communicating with one another? What are the signals that are of interest? It's an important thing to figure out. Now, I'm not going to get into too much detail about that, but let's just tell you that, say, okay, let's skip over that as well. If you were trying to figure out what's going on inside some silicon chip that you don't know about, you know, you've stolen this from your competitor's lab, it's a great chip that does something, you want to figure out how it works, then one of the things you can do, because you know that what you're mostly interested in is what sort of bits, what sort of ones and zeros does it produce on the output, which is levels of voltage, depending on the input that goes in. Well, those are the signals you're going to look at. You're not going to look at how this chip heats up the environment around it or how cosmic rays affect it or anything like that. You're interested in those signals. And sometimes to simplify the problem, you can open up this chip and you can sort of look at its surface and see what's connected to what, and it helps you get there a bit faster. I'll get into that in a second. So first of all, in the brain we also have some notion of what kind of things are the most important signals. Now, we don't really know what the most important ones are, of course, because like every other real object in the universe, they're affected by every signal that you could possibly think of. I mean, there are four main ones in physics that I have on a previous slide, but I'm not going to go through that. But if you break it down into, say, other things like just various electromagnetic radiation, etc., any object can be affected by, say, pressure, by all those different types of effects that exist in physics. Neurons as well. Neurons can fire because they receive an electric signal, or because they become activated by light, or because they become activated by sound, by vibration, by pressure. All those things are possible, but that's not usually how it works. So first you want to look at how do they normally communicate with one another. And it so happens that we do have something called spikes, that's these activations of neurons, that tend to be really important for sensory data. It all gets transferred into spike trains, as well as muscle output, that's also spike trains, and learning, because it's really the activity at a neuron and another neuron, and when they occur, so the timing of those spikes, that determines whether a connection is strengthened between the two neurons. That's learning. So spikes seem to be one of the most important signals to look at first. Okay, I think I already just said this. So if we could predict, say, the spike times, the times at which neurons spike in the brain at this moment, and then the next time when spiking occurs, this neuron, that neuron, that neuron, if we could predict all the spike times, there's a fair probability that if you could do that, you would already have a basic whole brain emulator. You would have something that operates just like the mind, because you're predicting all these spikes as they're supposed to occur. It's basically producing the same output. Now that doesn't mean that that's the end of everything, of course, because you have to do this kind of research in cycles. And if something isn't enough, if it's not producing the right output, it means you need to find where something's missing, add new signals, or add new components. Okay, now, even if we know what the signals are, it's still a pretty huge problem. You've got a big black box, you can't just look at what are the output spikes to the action, to say the actuators like muscles, and what are the input spikes, the sensory input we're getting, and so that doesn't tell us how we can figure out everything that's inside the brain. It's a huge combinatorial problem. There are so many billions of neurons in there that really it's an over-parameterized system. You can think of so many ways that the input can produce the output if you only have a little bit of it to look at. If you haven't watched the system since birth, there's so much you could be missing in there. So that's not what you do. You try to break down these problems into manageable things. So, for example, you can simplify the system by breaking it down spatially. You can take every neuron in the brain and break it down into what we call a compartmental model. That's this thing that you're seeing right up here. It's basically you've taken the neuron and you're breaking it up into such that every little piece, every tiny piece of these dendrites, these little branches that connect neurons to one another, is all broken up into little cylinders, and each cylinder represents an electrical circuit analogy, and there are Hodgkin-Huxley equations that determine how the sort of the ion channels in there operate and how this makes the electrical activity flow between them, so that you can get an idea of, well, if a spike arrives from a synapse into a dendrite at one point, what's going to happen to this neuron? You can figure that out if you have a model like this that's been broken apart into all those compartments. So now, for each one of those simple compartments, the system identification problem is much easier. If you can actually measure what's going into that and what's coming out of it, it would be solved if you could do that for each one of those. Of course, there are a lot of them, And it's not that easy to measure things inside the brain. So this is where the real work starts. After you have an idea of how to tackle it, you need the right tools. And that's really what, so basically what I do is I make sure that I find all the people who are working on the various kinds of tools that could apply to this, or the new tools that we need to make, so that this becomes a reality. And I've recently been joined by the 2045 initiative. That's a somewhat larger organization that sort of seeks the same goals. And so now we're able to build a roadmap and to create the network of scientists and projects that are necessary and additional funding for this. But I'll get into that in just a second. I just want to show you what this roadmap looks like. So here we have basically whole brain emulation, and there are 4 main pillars of what needs to be done. So you need to break it down into simple system identification problems, meaning let's look at lots of little pieces and try to define what's going on in each one of those. We call this the structural connectome problem. So that's try to look at it as a big structure with lots of little pieces, break it apart like that. Then you need to be able to characterize what's going on in each of those parts. Just looking at them visually gives you some idea. You can get an idea of, say, if this dendrite is yay thick, then it means that it probably has a resistance of so much. So you can get some idea of the parameters that you need, but it's not really very reliable if you're going to do that for millions of little pieces that you're measuring, where there are measurement errors that could be accumulating, and where there are parameters that you may not even be able to see. So ideally, you want to be able to record from each of these neurons and get at least a reference shape of what kind of activity it has. This gives you a characterization that you can use. So that's where the functional connectome comes in. This is the characterization of all the parts. And then you need something to emulate on, so some platform that would run this. That's actually not a huge problem right now. It certainly isn't at this moment, and looking at how that's developing, I don't think it will be in the foreseeable future either. I think that's always going to stay a step ahead of building the tools to get the data. And then we have this other 4th pillar, very important. It says there, validation. It's basically the iterating over the other 3 pillars, doing experiments, proof of concept, to test whether the hypotheses that you have are correct, find out which parts are not working as well as you'd like, and then improve each of those 3 pillars. So it's going to go through a few cycles of that. And on the bottom, you already see that there's some projects that are linked in to each one of those. And I'm going to say a few words about that as quickly as I can, seeing as very little time. Now, for the structural decomposition, you can go about that in a number of different ways. You could look at voxels. Those are little cubes, little areas inside the brain that you would look at, for example, with an MRI. Now, this is very rough. Using an MRI, that doesn't give you a very small voxel size, so you're not really decomposing the problem much. Further, you could look at how cells are connected to one another using the activity between them. If you have them, say, stuck on an array of electrodes, and you could look at when they're active, and then from the activity and seeing if neuron A always activates before neuron B, then there's probably a connection between them, so you can infer it that way. It's also not very effective at large scale. Much better is what Anthony Zador came up with, which is using RNA, DNA tags. Basically, they are unique tags that give you an identifier. You make sure they enter a neuron and that they go right across the boundary from one neuron to the next using the synapses as a conduit. And there, if you pick them out, you've got a label now telling you that the tag, the DNA tag from this neuron, somehow reached the other one. So as you look into each of those neurons and see which tags arrive there, you basically have pointers leading to all the other neurons that are connected to it. That's a biological way of figuring out this connectome. And then probably the most promising at the moment is this making stacks of electron microscope images, which I think I'll show you a little bit of. Basically, the concept there is to take a piece of brain, put it in something as solid enough to cut, then make very thin slices, put it under an electron microscope, and look at those electron microscope slides basically to come up with the structure of what was there. And let me try to see if I can do this for you. So after, okay, so this is basically, oh, it's not running on there, it's running on this other side. It's not good. Well, I don't know how to fix that problem right now. Oops. So let's just get right back into it.
Okay. Linux.
Yeah, that's not gonna be that easy. That's weird. I tried it on a monitor yesterday and it worked, but I guess it's not always the same. This is unfortunate because it's a very nice example of how you make slices from the brain and then what it looks like when you identify structures in there. Maybe one of the other videos will work. Okay, so almost there. This one was the one that's not working, unfortunately.
It's actually crashing.
No, it's not crashing. It's still here.
It's just not showing.
It's not showing there, yeah.
Is it a mirror? Yeah, mirroring and split.
Yeah. Actually, it is kind of strange because now it's not showing up anymore at all. So I can't seem to skip past that.
You said it's a mirror display, so you only have one display. That'll help.
Well,
There's a way to do it.
It doesn't work that easily.
On the system level, when you have a display control, just set it to be mirrored at your preferences, like operating system level. Just take away the two displays.
Well,
All displays.
Yeah, I tried this yesterday. This one didn't really work actually.
It's the only way it's going to
No, that's not working. Okay, well,
go back. Yeah. So, do without the visual, it's okay.
Can you drag the video to the Apple screen?
maybe I can actually. Let's see. That might be possible. Yeah, that's a good idea. Let's see if it shows up over here. Okay, so we've got this popping in here.
Dragging, yay.
Yay. Oh, wow,
But don't
just
resize
popped right
it.
back.
Just minimize it again. That's it. And just play from there.
Okay. Well, let's see if that works. Where am I? I can't see what I'm doing. Yeah, so how do you... Get down here. Okay. Well, it looks like I'm not going to get time to get into any more of this. Basically, what I was trying to show is just that there are indeed projects that are working on each one of those different aspects of how you would do whole brain emulation. There are biological tools as well as more typical electronic tools that we're using for that. So there are ways of addressing the problem of getting the whole large-scale data out of there, the structure, but there are now also emerging tools for the functional side. There's something called the molecular ticker tape, which is basically using DNA as a reporting mechanism inside each of the neurons and reporting on them when activity is happening. That's one of the most exciting new things that just came out of the George Church lab in Harvard. And I was there just 2 days ago to talk to him about how you could combine this with the scanning, and he told me that he has a new way of doing sequencing in situ in slices. So basically you can make the slices sequence to find the activity data that's being recorded on these molecular ticker tapes and still use that slice data to also make the structure, so they need both combined. And if it's true, and if both of those actually work together, then in principle we have the tools to do the first proof of concepts with small animals, like say for example the Drosophila fly or something like that. They work on pieces of that, or maybe retina in humans. There's already been some work in retina, so that's a good place to start. Anyway, I don't have any more time, but if you want to talk about this some more, then please come see me after the talk. Thank you.